Organoids, their advantages and flaws. A presentation on organoids, their flaws and advantages. Not to be confused with whole organ growth like that of the vacanti mouse. Organoids are small structures that can be grown in vitro. These small organ-like structures are smaller than standard organs and are meant to simulate the environment found in those organs. Organoids are a specialized version of a 3D culture. But while a square is always a rectangle, a 3D culture is not always an organoid. Organoids are meant to grow cells in a fashion that allows them to express their own extracellular matrix and organize themselves. This is supposed to allow for specificity in the cells, giving them a more organ-esque internal environment. These organ-like cultures can be used as test subjects without the risk of using human models, but also using human cells. An interesting and useful part of these cells is that they can be tailored to a patient by growing them from a patient's own cells. Organoids can be grown a variety of ways from a few different sources. Usually, they are grown from stem cells, but can also be grown from tumor cells when required. To grow an organoid, cells must be placed in a gel. This gel is then solidified in a 3D structure to allow for the organoid growth structures. From here, media is added across the outside of the gel to provide nutrients and other growth factors as desired. Organoids are then allowed to grow and enlarge until they are ready for use. In some cases, these newly formed organoids can be passaged and recultured to grow further samples for testing without requiring a new harvest of cellular material. While organoids seem like a great concept up front, they are still a relatively new technology in the field of culturing cells. With this, there are still many challenges to overcome. For example, while it is touted as a way to allow for similar conditions to that of an organ, it is not the same as an in vivo organ. There is no external pressure from surrounding cells, which can be a determining factor in organ growth and maintenance. Similarly, there are no chemical or neurological signals. If growing a muscle organoid, the in vitro organoid would be missing many of the inputs that would help create an actual in vivo environment. With these challenges already in place, organoid growth also requires special attention to ensure their continued growth, including the addition of other nutrients and chemicals to properly grow. Special needs for growth are an increased cost to any organization looking to use them in study or treatment. Now, you might be asking, if there are so many challenges to growing organoids, why would we use them with other methods available? Well, 2D methods have been used and studied in classes for years. Unlike 2D growths, organoids do not lack the cell-cell interactions that would come from having a self-organized group of cells. With this, chemical distribution more closely resembles that of an organ in these models. This factor leads to better simulations of how cells would actually respond to drugs or chemical inputs. These cultures are usually genomically stable, allowing for long-term growth and studies. Organoids also have the ability to be cryopreserved with relative ease, allowing future use for research or study long after their initial collection and growth. Outcomes from using organoids as models for treatment include many direct-to-patient benefits that could be applied with the current understanding and techniques. Organoid models can be used to highlight new pathways in organ function, not generally able to be seen in standard cultures. It can also show interactions in tumors leading to their growth, allowing for projection and possible drug targets for enhanced treatments. They can also be used to simulate gene activation in subject development or in that of host microbe interactions. Both areas being extremely difficult to study in vivo, organoids are extremely beneficial. The organoid culture model is far-reaching in implications and well worth the challenges still to overcome with them. I hope you've enjoyed today's presentation on organoids. If you are interested in learning more, these are my sources. Each have far more information than I could fit in a short presentation and are great sources for more information if you have interest.